Okay, so thanks, Nandini, for having me. Oh,
No, you got to start.
ah, there we go. Okay. Um, yeah, so I'm Chuck Pepe-Ranney. I work at AgBiome. We're actually just up the street. And AgBiome is an agricultural biotechnology startup specializing in the discovery and development of microbial solutions to crop pest and crop disease problems. And so you could say that we are in the microbiome business. And today I want to talk about a couple issues with microbiome data that make it very difficult to study microbiomes. And then I want to let you know how we at AgBiome are addressing those challenges. But first, let me give you a little overview of the AgBiome pipeline as context for some later slides. So our pipeline is pretty simple. We go all throughout the US and collect samples. And then we take those samples, and from them we isolate microorganisms. And then we take those microorganisms and assay them for specific activities that we're interested in. And then when we find microbes that we're interested in, we develop them into products. And so that's what this slide is depicting here. And like I said before, we're in the microbiome business, and that's really exciting because microbiomes have really captured the public's attention recently. And this attention has culminated in the Microbiome Initiative from the White House, right? So this is a federal initiative to foster microbiome science in the United States. Okay. So we're seeing a lot of headlines like this. Why your gut microbiome could hold the key to solving the obesity epidemic. And I think these headlines are really interesting, and it's really interesting to sort of dig into the science behind them and sort of try to quantify the uncertainty in that science. And if you take anything away from this talk, it should be that the answer to whether or not we can treat obesity by altering the gut microbiome is uncertain, okay? And what I want to do is tell you why I think that that answer is uncertain, and then how those aspects that make studying microbiomes challenging are being addressed at AgBiome. Right, so we're starting to accumulate a number of studies in the sort of microbiome science community. For instance, we've screened a lot of obese and lean humans and looked for links between obesity and the gut microbiome. And there are many individual studies that show really compelling evidence for some signal in our microbiome with respect to obesity. But when you sort of take a step back and do a meta-analysis of all of the studies in total, a lot of the signal is really actually quite weak, right, and not all that reproducible. And so, I mean, why is that, right? And I think the answer is really complicated, but I think there are a couple features of the microbiome that cause this uncertainty, in addition to many more, but I'm going to focus on two. So if you're really interested in this question of obesity and the gut microbiome, although this is a biotech conference or session, I would point you to this meta-analysis done by Pat Schloss's lab at the University of Michigan. And I think it's of interest to anybody that's in the microbiome science community, because it really helps us to get a handle on the uncertainty in multiple studies as opposed to just single anecdotes. Okay, so there's a couple items that I really want to focus on that pose challenges to the microbiome scientists. So the first one is that the microbial world is really, really diverse, okay? So this is a review of sort of the diversity of life as we know it on Earth that came out in 2015. And what you see are what you see here is a phylogenetic tree. So this is evolutionary history of all the organisms that we know to exist, right? And all of these tips over here in this tree, all of these clades are bacteria. We have this sort of clade of, this was sort of the big story about this paper, was that there was this giant clade of relatively unknown, newly discovered microorganisms. And then, in some of these like very limited clades down here, you have all the vertebrates, right, for example. So the point is that, humans and all the animals and plants that we can are tangible to us, that we sort of intuitively know and understand, they make up a very, very, very small slice of the diversity of life, right? Microorganisms are a huge and overwhelming component of the diversity of life, and that makes studying things like the gut microbiome, agricultural microbiome, whether it's soil or rhizosphere-associated soil, crop endophytes, et cetera, very difficult. But this diversity is sort of multi-dimensional, okay? So even in relatively narrow groups, like a microbial species, there's tons of microdiversity, okay? So this is a study done by a group at MIT where they screened the genomes of 200 or thereabouts strains of a single microbial species called Vibrio splendidus, okay? And what they found was that 180 of those 200 genomes, all from this one species, which you might expect to be fairly uniform in its genome content, were unique, right? So, it's almost like, and I think they make this this comparison in the study. It's almost like these genomes are like snowflakes. Each one is unique. Okay, so we don't, only have this really big diversity in these broad groups. We also have lots of microdiversity. Okay, and both of these dimensions of diversity are really starting to dawn on microbiologists and make it very difficult to study microorganisms. Okay, so the second thing, beyond diversity, which makes microbiome science challenging, is that we don't know what a lot of these microbes do. Right? We don't know their activities. So our ability to detect and also detect the presence and absence of a microbe in the environment has far outpaced our ability to assay microbial activity. And really the best way to know what a microbe does is to grow it in culture. And so this figure is showing how little microbes we have in culture. So on the y-axis here, we have different divisions of bacteria and archaea. And on the x-axis, we have percentage of kinds of microbes in those divisions for which we have microbes in culture, right? So for instance, we have only about 20% of the kinds of Bacteroidetes that we know exist in culture, right? And the other 80%, we haven't grown in the lab, and we haven't actually observed directly their activity. So we've got this like overwhelming diversity, and much of it is understudied with respect to function, with respect to physiology, ecology, activity, et cetera. Okay, so very challenging to study the microbiome. And at AgBiome, we're trying to address this diversity problem, specifically the microdiversity issue, as well as the sort of lack of activity information for all these microorganisms. So the first thing that we do is we sequence the genome of every microorganism that we isolate, okay? So this is important because many of the methods that microbiologists have used to observe microbes in the environment are fairly low resolution with respect to that microdiversity, right? So this genome variation in those Vibrio splendidus, for instance, that we talked about earlier, that would be sort of invisible to some of the methods that microbial ecologists are using. But AgBiome, we want to see that microdiversity represented in our database, right? So we sequence the genome of every microorganism that we isolate, and that gives us this high resolution information of all of our isolates. So this is just a heat map of about 40 strains in our collection that would look identical to many of the methods that microbial ecologists use. So on the x-axis, we have genes. Dark colors represent the presence of a gene, light colors the absence. And although these microorganisms would look fairly uniform to some methods, they actually are quite different from one another, right? And so we're capturing this microdiversity in our collection, and we can really get at high resolution relationships between microorganisms. Okay, so that's one way that we're tackling that challenge. And just for fun, I just wanted to give you like a quick glimpse at our database, or a peek at our database here. So we have nearly 30,000 microbial genomes in our collection, and it's continuously growing. And in this figure, each dot, small dot, represents a genome, and genomes that are related to each other in this high resolution comparison between genomes are connected with an edge. And then the genomes are colored based on their phylogenetic affiliation. So these blue genomes here are from a genus called Arthrobacter, which is a very common soil microorganism. But you can see that even within this Arthrobacter genus, we have in our collection lots of different maybe ecological, really sort of like units that are defined by genomic content, okay? So let's zoom in here. And we can hover over the points. And the other thing that I want to make clear is that for every single genome, we have all this metadata that we can leverage to try to understand where to find microbes that are of interest for biotechnology, and really just like searching for patterns that we can leverage. So if this is a Pseudomonas, we know that the species and where we isolated it from and what crop it was produced, or the sample was taken from. And so we have just lots of information that sort of looks like this, okay? And we have all these really sort of interesting shapes and relatedness patterns that we're making sense of. Okay. Okay, so coming to the end here. So the other issue that we talked about was this issue of activity and function and unknown function. So, one of the ways at AgBiome that we address this is simply by culturing microbes, right? So we're only interested in things that we can grow in the lab with respect to the biological solutions that we're producing. Okay, so that's one part of it. So we have all of these microbes in culture. We can observe their activity. We can observe their function, as needed. But the other thing is that we've assembled a team, sort of a multidisciplinary team with the expertise to produce assays that are rigorous and reliable and high throughput, right? So we're a microbiome science company, but microbiologists are just a small fraction of our employees, right? We also have computational biologists. We have entomologists, right? Because a lot of our activities have to do with the relationships between microbes and crop insect pests. We have people that understand field trials, how to interpret and conduct field trials, et cetera. So to really get at microbial activity takes an interdisciplinary team and to develop assays. And that's how we are sort of bridging this gap between identity of the microbes and their function. All right, and with that, I'd like to take any questions that you might have.